Hello everyone, this is Dr. Ruby and welcome to the Module 4 lectures. This is part 1. It goes with your Chapter 10 in your textbook, Anatomy and Physiology of Muscle Tissue. In this chapter we will focus on the anatomy and physiology of a skeletal muscle. The two major tissue types, skeletal muscle tissue and muscular fascia, will be examined. On the next two slides you will see the objectives for this chapter. Again, I encourage you to use these objectives to study so that you are prepared for the quiz. An important part of studying and knowing this information is to know the key terms and words. So if you come across a word or term that you don't understand either in the lecture or the reading, make sure that you look it up. And if you haven't subscribed to the textbook resources, there's a really nice section there that goes through the terminology that's free to you with your textbook. So if you haven't accessed that yet, you may want to. Again, here's more objectives. There are three types of muscle tissue in the human body: cardiac muscle tissue, smooth muscle tissue, and skeletal muscle tissue. Skeletal muscle is striated in appearance and it is under voluntary control. This illustration really shows the big picture of muscle contraction. In B, the muscle has contracted and shortened, pulling the bones of the forearm toward the arm. There are two major types of tissues in a skeletal muscle. There's skeletal muscle tissue and fibrous fascia connective tissue. The skeletal muscle does the work, and the fibrous fascia tissue provides the framework. Muscles do also contain nerves and blood vessels. This is a cross-section of a muscle attached via its tendon to a bone. It contains both muscle tissue and fibrous fascial tissue. This fibrous fascial tissue provides the framework in which the muscle does its work. A muscle cell is the same thing as a muscle fiber. These fibers may vary from a ½ inch to 20 inches in length, and these muscle fibers are bundled together in what we call fascicles. This diagram shows three different ways in which muscle cells can attach to one another and be oriented within a muscle. In A, long muscle fibers lie parallel and run the length of a muscle. In B, shorter muscle fibers lie parallel and end to end. In C, muscle fibers lie parallel and overlap. In this illustration, you can see the fascicles. Each fascicle is a bundle of muscle fibers that are separated from adjacent fascicles by fascia. Muscular fascia creates the structural organization of a muscle. The name of it depends on its location. It could be called endomysium, perimysium, or epimysium. If it is round and cord-like, it's a tendon. If it's flat and broad, it's an aponeurosis. Some textbooks use the phrase myofascial unit to replace muscle. One of the advantages of this approach is that it combines the muscle with its fascia. Pay attention to the figure above on the right. Note the large number of myofibrils that fill the sarcoplasm of the muscle fiber. These myofibrils run the entire length of the muscle fiber. A myofascial unit is a term for a muscle that specifically includes the tendons and or the aponeuroses of the muscle, as well as the endomysium, perimysium, and epimysium. Myo refers to the muscular tissue component, and fascial refers to the fibrous fascial component. Cellular structure names are similar to other cell types, but they contain the prefix sarco. Cytoplasm equals sarcoplasm. Endoplasmic reticulum equals sarcoplasmic reticulum. Cell membrane equals sarcolemma. So sarcomeres are small units that make up myofibrils. Sarcomeres are laid end to end from one end of the myofibril to the other end. Muscle fibers contain myofibrils. They run longitudinally the length of the cell, and there are approximately 1,000 myofibrils per fiber. This is the structure of a sarcomere. Note the thick myosin filament at the center of the sarcomere and the thinner actin filaments attached to the Z-lines. Here are the objectives for section 10.2. The sliding filament mechanism explains how sarcomeres shorten. A message is sent from the nervous system. The sarcoplasmic reticulum releases calcium into the sarcoplasm. Calcium ions then attach to the actin filaments. Another name for the sliding filament mechanism or theory is the ratchet theory, since the mechanism of muscle contraction bears some resemblance to those of a ratchet wrench. The myosin heads attach to the binding sites on the actin filaments. Then the myosin cross bridges bend, pulling the actin in toward the center. Cross bridges break, reattach, and bend again. This illustrates the steps of the sliding filament mechanism. It begins when binding sites are exposed and ends when the process begins again. If the cross bridges succeed in bending, the sarcomere shortens toward its center as the muscle contracts. When the sarcomere is fully contracted, it's at its shortest length. The energy source for a sliding filament mechanism comes from ATP. The process is quite simple. There's stored ATP. There's regeneration from creatine phosphate, regeneration from anaerobic breakdown of glucose, and regeneration from aerobic breakdown of glucose. Aerobic and anaerobic mean the absence or presence of oxygen. Aerobic is when oxygen is present in the chemical breakdown, and when anaerobic is when there is no oxygen present. The energy source for the sliding filament often requires oxygen debt, the debt of oxygen that is needed to convert the buildup of lactic acid back to glucose. Oxygen debt is the reason we continue to breathe heavily after exercising vigorously. Our bodies need the extra oxygen to convert lactic acid created by the anaerobic breakdown of glucose back into glucose. The nervous system also has control of muscle contraction. The muscle fibers cannot contract on their own. They require instruction from the nervous system. The nervous system has control through the neuromuscular junction where neurons meet muscle fibers. The muscles cannot contract on their own. They require signals from the nervous system. This signal happens at a motor end plate that is part of the muscle fiber's sarcolemma. It is specialized to receive neurotransmitters from the nervous system. This is an illustration of the neuromuscular junction. Neurotransmitters from the motor neuron are released into synapses where they attach themselves to the motor end plate of the muscle fiber. Once the neurotransmitters are bound to the motor end plate, an electrical impulse travels to the interior of the muscle fiber via the transverse tubules. These are also known as the T-tubules. When a motor neuron reaches a muscle, it branches numerous times to synapse with a number of muscle fibers. In this manner, a single motor neuron controls a number of muscle fibers. All motor units contain one motor neuron. In this photo, this particular motor neuron has branched out to control 4 muscle fibers. The number of muscle fibers in a motor unit varies from as few as 2 to 3 or as many as 2,000. This drawing indicates that since a muscle contains multiple motor units, they need not all contract simultaneously. However, when a single muscle fiber is contracted, it contracts all the way to 100% of its capacity. Motor units cannot have a partial contraction. It's an all or none response. Here are the objectives for Lesson 10.3. Now we will look at the sarcomere structure in more detail. The letters A in A band and I in I band stand for anisotropic and isotropic. These are simply terms that are used to describe how light is refracted. Figure A here shows one myosin filament surrounded by 6 actin filaments. Figure B shows how multiple sarcomeres that lay next to one another are arranged. Figure C is a cross-section of a sarcomere. In the top diagram on this slide, you can see that there are 3 sarcomeres laid end to end. In actuality, there are 10,000 sarcomeres laid end to end in every inch of a myofibril. The sarcomere structure is made up of the myosin filament and the actin filament. Many myosin molecules bind together to make the myosin filament. They each have a myosin tail and a myosin head. In the actin filament, there are 3 separate protein molecules: actin, troponin, and tropomyosin. The tail is the main length of each myosin molecule, which incidentally resembles a golf club in shape. You can see how multiple myosin molecules are attached to one another to form a myosin filament in this diagram. Tropomyosin molecules normally block the binding sites of actin. Troponin molecules have the ability to move tropomyosin out of the way so that the binding sites of actin are exposed. This occurs when calcium is present. The sliding filament mechanism has 4 basic steps. In step 1, the ATP molecule attaches to the myosin head. In step 2, calcium binds to the troponin molecule. In step 3, the myosin head attached to the exposed binding site of the actin filament. And in step 4, the myosin head bends, pulling the actin filament toward the center of the sarcomere. In this manner, a sarcomere shortens. If all of the sarcomeres of a myofibril shorten, the myofibril shortens. If all the myofibrils of a muscle fiber shorten, the muscle fiber shortens. When a number of muscle fibers shorten, the muscle shortens. This is how a concentric contraction works. There are both red and white muscle fibers. Red fibers are red slow twitch muscle fibers. White fibers are white fast twitch muscle fibers. There is also a third category, which we call the intermediate twitch fibers. Although some conversion seems possible, for the most part, the ratio of red and white fibers in our bodies is genetically determined. Red fibers contract more slowly, white fibers contract more quickly. Every muscle has a mixture of red and white fibers. Myofascial meridians and tensegrity. Under the myofascial meridian theory, muscles operate within continuous lines of fascia that span across the body. This places muscles in a larger structural pattern. When a muscle's tendon attaches onto a bone, although most of its fibers usually do attach onto the bone itself, some of its fibers are continuous with the fascial fibers of an adjacent muscle's tendon. Figure A on this slide shows the superficial back line myofascial meridian, and Figure B shows the superficial front line myofascial meridian. Figure C shows the lateral line myofascial meridian, and Figure D shows the spiral line myofascial meridian. Figure E shows the superficial and deep front arm line myofascial meridians, and Figure F shows the superficial and deep back arm line myofascial meridians. Figure G shows the back functional line myofascial meridian, and Figure H shows the front functional line myofascial meridian. Figure I shows the deep front line myofascial meridian. The drawing on the right shows a myofascial meridian in a monkey hanging from a tree. The photo on the left shows a dissection of the superficial back line myofascial meridian of the body. Conceptually, if a strain occurs at one point along this myofascial meridian, effects of that strain could be felt in distant locations along the myofascial meridian. What are the origins of the word tensegrity? Well, the term tensegrity comes from the phrase tension, integrity, and it was first used by a designer named R. Buckminster Fuller. Tensegrity means the structural and functional integrity of the body comes from tensile or pulling forces. If you look here at the diagrams, and on the left you can see the tensegrity, the compression members or the bones suspended by the tension elements or the myofascial meridians. Now to the right you see a diagram. This is a common representation used to teach tensegrity. It's a model that has some give to it. You've maybe seen them, you can see them, they're toys sometimes where they have some give to them, but they have a solid structure. So this is just a model of what we think of when we talk about the concept of tensegrity. This concludes the first lecture for Module 4.